Yeah, that's the. Okay, excited to have the November Molecular Connectivity Working Group seminar, and people who are not in the Molecular Connectivity Working Group, welcome. Also, all members, welcome. As I said earlier, we will repeating here a version of an OHBM symposium that happened in Brisbane, unfortunately under some bad sort of funding sort of malaise where many of us couldn't make it and there was a lot of uncertainty. So this is the event with the almost the original lineup of speakers. And so we're going to talk about validating brain connectivity measures, as it said in the flyer. And we look at several kinds of validation, and the order of the talks will be like in the symposium flyer. We'll have Alan Jasanoff from MIT talking about biological validation. We have Daniel Kristanto standing in for Andrea Hildebrandt, who couldn't be here, talking about multiverse analyses and the impact on connectivity measures. And then Zhen-Qi Liu from McGill will talk about statistical properties of connectivity after a very comprehensive and interesting survey that was published. And lastly, we have Matej Perovnik, and hopefully he can make it here, and his clinical duties won't prevent him, talking about applications and extensive validation of PET brain networks that were derived with the scaled subprofile model. So I don't want to take a whole lot more of your time. I would maybe invite my co-host, Daniel Talmasov, just give a very brief introductory remarks. Daniel, take it.
Thank you, Chris, and welcome everyone. Good morning or good afternoon, depending on where you are. Just a very brief comment to contextualize the conversation that we're having as a part of our work and ongoing work in the field. This really came out of an ongoing conversation within this group and with our colleagues internationally regarding our work on validating considering how to validate brain connectivity studies that use molecular imaging techniques. With regards to this, we're talking about molecular connectivity studies, of course, extending beyond anatomical connectivity or even beyond functional connectivity that has become popular using resting blood oxygen-dependent MRI measures that infer statistical dependencies between regional activation to statistical dependencies between molecular imaging measures taken across subjects or within subjects in brain readings. This culminated in this symposium in Brisbane, which, as Chris mentioned, many of us, including many of us in the US, were precluded from coming at the time. And so we're happy to gather with you all and have a chance to participate and hear these talks again and sort of reconvene. As we have been thinking about validating molecular connectivity in the context of brain imaging, we have sort of divided into three areas, and I encourage everyone to sort of think about how these areas maybe coincide and cohere as we hear the various talks, be they statistical, biological, or clinical forms of validation, with statistical validation referring to sort of consistency and robustness of measures taken and analyses taken using molecular imaging methods that infer connectivity, biological validation referring to two forms, really, one being cross-comparison with other neuroimaging modalities, and the other cross-comparison to reference standards, laboratory standards such as microscopy or staining, or indeed other time-dependent measures and clinical physiology measures. And finally, of course, clinical validation with implications for disease monitoring and treatment effects. That's all I want to say. I'm glad that we're able to gather together and have this conversation online now, as well as in person previously. And I think I'll turn it back over to Chris or Arianna to begin introducing our first speaker.
Yeah, Arianna, maybe take it away and we could start for real.
All right. So, well, I think, Alan, the mic is yours. So we'll start with the biological validation. and in particular, how a genetically encoded activity reporter in animal models can inform the biological basis of fMRI brain connectivity. So welcome and looking forward to discuss.
All right, thanks very much. And I'd like to thank Chris and Shin, Daniel, and you, Arianna, for hosting. So I'm going to share my screen here. And let's see, I think this is the one I need. Here we go. Can people see this? I saw at least one thumb. That's a good sign. So just a quick caveat. I was one of the absentee presenters at the Brisbane meeting, and I have updated my talk since then. So for those of you who saw the recorded talk, hopefully there's something new. But the topic is the same. It's brain-wide analysis of functional connectivity and its relationship to physical connectivity using molecular MRI. By the way, I'm a neuroimager, but I'm also in the Department of Biological Engineering. You'll see that there's a bit of fusion of these two things in my talk. So I think a premise that we all agree on is that network level function in the brain depends on the connections in the brain. And, there are a number of outstanding questions about how these connections function to organize the various aspects of brain activity and ultimately cognition and behavior. So maybe utmost among these questions is, well, what information do the connections transmit? How is transmission across projections in the brain related to other connectivity and activity measures? And that, in a sense, is what this little meeting is about. And then how do patterns of information flow help explain network-wide function? And ultimately, of course, the big questions about how the brain works. These questions are very difficult to answer. And, it's going to be exciting to hear how the different speakers here are addressing it in different ways. I am someone who works with molecular level imaging tools and model organisms. And I know that the dominant ways to address these questions in the sort of basic neuroscience community that I'm in involve working with transparent organisms where we can see every cell and their activity and then chop them up afterwards and look at how they're connected. And even in, a low-level animal like a mouse, well, it's not that low level. It's actually very, very, very difficult to address these types of questions. And that's a challenge that my lab has taken on, both because we're interested in the questions and because we work on tools that can be appropriate for this. And so I'm going to tell you about our work that combines whole brain imaging, brain-wide imaging with MRI, with a tool that we've developed, a genetically encoded activity sensor for fMRI that we deliver using viral tracers in animals. So it enables us to combine a molecular or cellular level readout, really, from the genetic tool with a brain-wide view of the animal and what it's doing, or at least what its brain is doing. All right, so let me explain what the tool looks like. We have developed this genetic activity reporter. It's something called a Gnostic. And it is a protein that's delivered to cells using a gene. So genetic tools like viral vectors can be used to introduce the protein and get it to be expressed inside neurons that we target with the virus. And when those cells become active, there we go. When the cells become active, calcium fluctuations in the cell trigger the activity of the protein, which is a variant of nitric oxide synthase. The protein thereby produces nitric oxide, which is a potent vasodilator that induces local vasodilation, and that in turn triggers an fMRI signal, a bold signal, very much like the standard bold signal that we see in ordinary fMRI experiments. So basically what it is, what Gnostic is it's a tool for converting activity of targeted cells into specialized fMRI signals. Now, in the ideal world, we would install our tool into whatever connections in the brain or cell populations in general we might be interested in studying. And they would signal their local vessels, and we would be able, in the pattern of neural connections that are labeled with the tool, we would be able to measure the activity of those cells directly. And we can achieve this, but it's through a little bit of complexity. The complexity arises because when we see our specialized bold signal, it's superimposed on the endogenous bold signal that comes from basically every cell in the brain that's activated during the stimulus or behavior that we're looking at. So we see a superposition of the gnostic and intrinsic signals from throughout the brain. Gnostic here I'm color coding with cyan. Intrinsic signals I'm giving this kind of lilac or purple color. Now, fortunately, we have a way to disambiguate those signals, and that's essential to how we constructed our tool. We use a drug called 1400W that will appear later in the talk to selectively turn off our gnostic, our activity reporter, while preserving the other signals in the brain. And so what that gives us is a more pure, just conventional fMRI image or data set. We can, that reflects the activity of all of the brain other than the cells that we've labeled. Well, actually, even the cells we've labeled contribute, but not at a special level. So the trick that we do is we do imaging in the absence and presence of this drug. And then by subtracting these two data sets, we get back our pure targeted cell activity only data set. That comes from the cells that are expressing or reporting. All right, so this is the tool that we use, and I think it's the first activity reporter for fMRI and probably still one of a kind, I think, in this space. And now I'm going to tell you a little bit about how we worked with it. And so let's ask the question, how we can use this gnostic to measure projection-specific brain activity. And the first study where we took this task on was published a few years ago, and I'll tell you about the experiments that we did there. Specifically in that study, we were interested in looking at activity in the reward network. And we applied our tool using a viral vector, herpes simplex viral vector that is transported retrogradely, so presynaptically, to sites that provide input to wherever it is that we injected the virus. We injected it into the striatum, and so we labeled inputs to the striatum, and we then examined the activity of those inputs under a rewarding stimulus that we could give in the brain of a rat. So that's where our model organism here is a rat. And so we performed the imaging in two ways, as I explained on the previous slide. So first, in the absence of this drug, 1400W, and then in the presence of the drug, we turn off our projection-specific signal, and we see only the background, or at least that's the idea. And then the difference reveals properties of the projections. And so when we did this experiment, oh, by the way, let me just note that, it seems cumbersome that we have to do two imaging experiments to get our projection-specific data. But actually, we get something extra for that because we, of course, also map out functional activation in response to the stimulus, in this case, the rewarding electrode stimulus here in LH. We get two things for the price of two, as opposed to one thing for the price of two. All right, so we did this experiment. And we saw different signals. So these are the signals that correspond to the gnostic projection-specific signal only. And you can see that there are these peaks of activity that are distributed at various spots throughout the brain. This is during the rewarding stimulus. And this provides some indication that the probe is working and that it's giving us activity measures of inputs to the site where we injected the virus, which was about over here in caudate putamen. Because this was the first study of its kind, we also did control measurements with the same exact layout, but using a virus that encoded M-cherry, a sort of neutral, boring fluorescent protein, rather than something that could be visualized by fMRI. And you can see that although the data aren't entirely clean, they lack these, most prominent peaks of activity that we could see here, over here on the left in the difference signal. And if we look more closely at the regional signals in the gnostic condition over here, in the absence and in the presence, of 1400W, you can see that there are indeed systematic decreases in the signal when we add this drug, and that reflect the specific contributions of the activity probe in these structures here. So what this tells us is that we're able to see consistent signatures of input to the striatum, the area that we injected with the virus, from diverse brain regions. So now, what can we learn from this tool now that we've identified that it seems to be functional? Well, one of the first questions that we asked is projection-specific activity that we record during during application of the Gnostic and reward stimulation, is it simply proportional across ROIs to the other various measures that we commonly use to judge connectivity? And so we looked at a few different measures. So the first and easiest was non-specific fMRI. In other words, essentially the fMRI data that we recorded after turning off our probe with that drug, 1400W. And we noticed by looking at the ratio of the Gnostic signal to the non-specific signal that there was quite a lot of variation over different regions of interest, different brain structures, in the relative amplitudes of these signals. So it shows us that we're getting distinct information from the projections. Now, possibly more interestingly, we also compared our measures to resting state functional connectivity to the striatal site where we injected our virus. So with the Gnostic, we're looking at activity of inputs, physical inputs to the striatum, whereas with resting state, of course, we're looking at correlations between distal structures and the striatal activity. And again, we saw pretty sharp differences between the Gnostic and the resting state amplitude. So these are ratios of the Gnostic amplitude to the, I think it's the Z-score from the resting state correlation here. And then lastly, we looked at another obvious measure, which is the physical connectivity as judged outside the scanner. And in fact, outside the animal's life, we took the animals, we sacrificed them, and we examined by histology where the Gnostic was expressed. And of course, the brighter expression areas are the areas that, transported more of the virus and presumably are more physically connected. But because the Gnostic is an activity reporter, of course, it doesn't necessarily track the pure physical connectivity between structures in the brain. And in fact, when we looked at an analogous ratio to the ones that I've looked at over here, we saw, again, quite significant variation in the ratio of transport to activity. So for instance, the structure SN, substantia nigra, is very densely traced, but not nearly as activated relative to some of the other structures that we looked like here. So we've been able to perform a dissociation between the information that the Gnostic is giving and these other forms of connectivity, of judging connectivity in the field. In addition, we can sort of take all these measures along with our Gnostic measure and put them together into sort of an informal circuit diagram that tells us which projections, these are all regions in the rat brain for those of you who aren't used to looking at this, we can see connections that are activated during the stimulation, this pink thing here, as well as regions that are activated, like, for instance, cingulate cortex or S1 over here, that are activated but not providing projection to the target site. They nevertheless show functional connectivity, that's the gray, and we can start to speculate about how information flows through the system via this structure, the caudate putamen, where we labeled. All right, so Gnostics provide distinct information from the other connectivity measures, and they contribute to circuit-level descriptions. Now, the dissociation that we performed up here told us how these signals vary among regions of interest. But there's a common perception in the, let's say, higher-level systems neuroscience world that the information that flows over connections should track the information present in the source regions. And of course, that must be true in some way. But is it true at a population level? That assumption is often made, for instance, in techniques like dynamic causal modeling or when we use GLM analyses. So we realized that we could look at this directly, and we did this in a different experimental paradigm. Here, we looked at responses triggered by a somatosensory stimulus to the forepaw of a rat. And we delivered our Gnostic to a structure in the thalamus that is densely connected to cortical and subcortical regions in the somatosensory system. This is a region called posterior-medial thalamus, or POM, and that it's analogous, for those of you who care about the visual system, it's analogous to the pulvinar, but in the somatosensory system. And this is unpublished work that hopefully will soon be submitted, but I'm eager to talk about it, so I hope you don't mind. Now, we looked at this system under more than one condition that allowed us to parse apart activity profiles of the Gnostic labeled projections and the non-specific regional activity here. And specifically, we looked over different stimulus frequencies that are known to elicit different response amplitudes as a function of frequency, essentially tuning curves, albeit only at 3 points. And we looked at these in rats, and we asked, to what extent do projections track activity at the source regions? And there are various different ways to look at it, but I'll show you one. Here we're looking at the preferred frequency as a map in the 3 slices of the brain that have the strongest responses here. And I think you can see by comparing the Gnostic projection-specific activity and the intrinsic activity, both measured in the same animals at the same time, that there are actually pretty clear tuning differences. So in particular, the Gnostic signal seems to prefer these lower tuning frequencies over here versus higher tuning frequencies preferred for the intrinsic activity. And of course, the difference in tuning varies by region, but I think you can see by looking at the map, at least, that there's a pretty clear distinction. We also saw differences in response duration. What about the opposite? If we look at projections from an individual site to 2 different sites, do those projections transmit the same tuning information? We could look at that actually in a single set of animals where we labeled, let's say, a representative brain region that projects, that gets input from different regions by stimulating both forepaws. So in one case, we're looking at the contralateral forepaw, and the other, the ipsilateral, but the information is passing through the system in 2 different ways. And if we assume left-right symmetry in the brain, which is, of course, slightly questionable, but nevertheless we did it, we can look at this question pretty simply. And what we saw, and this is just one example over here, if we look at connections between one source region, here we're looking at zona incerta, and its projections to 2 different target regions, and here we're looking at contralateral POM and ipsilateral POM. If the signals going to the 2 targets was proportional, in other words, if the same information was relayed, we would expect all the dots here, which are the amplitudes under different stimuli, to fall along a line. But we don't see that. In fact, we see differences in tuning. So for instance, the contralateral POM is more tuned for the strongest stimuli, ipsilateral for this intermediate stimulus frequency here, and so on. So in other words, we see a dissociation between the information projecting to 2 targets. So what I hope you can see from this slide is that projections are tuned distinctly from their sources, their source areas in the brain, and also from each other. So 2 projections from the same source are tuned differently to different targets. Okay. Now we looked at, in a sense, the opposite direction. The previous slide was looking at how stimuli diverge from the population responses at their source regions. Now, how do they combine at the targets? To do this, we looked at time courses, which we could obtain over long periods of time, sort of 20-minute-ish. We could look at intrinsic time courses and these Gnostic time courses, which we obtained by differencing intrinsic signals in the presence of, by differencing fMRI signals in the presence and absence of this 1400W. And we asked whether we could notice systematic relationships among multiple inputs to a target structure. And we looked specifically at inputs to this area POM. We also looked a little bit at S1 inputs, but I'm not including that here. And the first thing we noticed is that when we do essentially functional connectivity, so looking at the correlations among all the inputs to POM from around the brain, these are all the regions, cortical in pink, thalamic in green, and other subcortical in gray. These are all the regions that show significant input to S1 under the stimuli we looked at. And you can see that they're all positively correlated. That's what the orange means. In many cases, equivalently strongly. The green outlines mean statistical significance over multiple animals. Now, if we look over different conditions, this pattern actually changes. So this is under contralateral stimulation, which gives, of course, the strongest responses because of the dominant pathways via the thalamus. If we give ipsilateral stimulation, well, we still see predominantly positive correlations among all the regions, but now the cortical response is less correlated with the other projections in the system. And under resting state, we see yet a different pattern where the thalamic regions, the thalamic projections, they're essentially intrathalamic projections, are all pretty strongly correlated, as are this projection from ZI, but the others are not as correlated. So these different conditions here clearly distinguish the pattern of information flow into the POM from the network of connections that we've labeled with our Gnostic and its viral vector. Interestingly, when we look at standard functional connectivity, okay, this is not all resting state. This is under 3 different conditions: contralateral stimulation, ipsilateral stimulation, and resting state. If we look at the intrinsic functional connectivity, we see a remarkably conserved pattern of functional connectivity. These are correlations between regional activities. That's consistent with, I think, an idea that many people have these days that, stimulus-driven activity and resting state activity shouldn't really expose great differences in functional connectivity, but it's clearly showing the difference between the measurement that's specific to projections and the measurement that is not. So we've learned that context, the stimulus environment, determines the organization of information flow. And we've also learned that the intrinsic functional connectivity, in other words, standard functional connectivity as measured from regular fMRI, is influenced by probably multiple signaling pathways that aren't limited to the projections that we probed with our Gnostic projection-specific measurements. Okay? All right, so the next question we asked is, are there functionally distinct roles for different projections? So we saw that they're all correlated, but that doesn't mean that the information coming over these projections is the same. And we looked at the simplest possible distinction between projections, which is, are they predominantly excitatory or inhibitory? And we can do this in animals because, of course, we can actually sack them and identify which cells are excitatory and inhibitory. So what we did is we looked at the rat brain post-mortem, and we compared the pattern of Gnostic staining in red in this histology diagram over here with staining for VGAT, which is a marker of inhibitory cells. So the green cells are all the inhibitory cells, not necessarily labeled. The red cells are labeled with our virus. And the inhibitory projections are yellow. They're the ones that are inhibitory and labeled with our virus. And these are projections to POM. So the injection site was somewhere around here. We can blow these up more clearly here. You can see that actually most of the cells around the injection site are excitatory. These are the labeled cells. They're not green. So we're assuming they're excitatory. Not all of them, by the way. Whereas in this other region, ZI over here, most of the cells are actually inhibitory. So the main input from ZI is an inhibitory one to this POM. And now we ask, well, are these cells doing the same thing, or is there some interesting distinction between how the excitatory and inhibitory inputs are combining in the network? And the first thing that we saw was just by looking at the various different Gnostic signals as a function of region in these different groups of regions here, is that basically all of the projection-specific signals that we looked at were either close to zero or positive. And that is agnostic to the presence of specific connection pathways. So for instance, from ZI, that's the one shown up here, also from this region called APT, anterior protectal nucleus, shown over here. These projections are mainly inhibitory, and yet they are sending information that tracks, for instance, the information from S1, the primary cortical activation site. And that's coincident with this, what we observed, and I showed you on the previous slide, which is that under contralateral stimulation, so the sort of the main forepaw stimulation, all these signals are positively correlated, whether or not they are primarily inhibitory or excitatory. And so I think what we're seeing here is that there is a balance between excitatory and inhibitory projection activity, a well-known EI balance, as it's called, but that we're able to observe it at a systems level, at the level of the entire network, by looking at these different projections here. Now, so obviously we don't imagine that EI balance doesn't matter for how the network works. So what roles does it actually play? Well,
Alan?
Yes?
Super sorry, it's a bit over time. I don't know if you can wrap up.
All right, well, this is my last data slide.
Thanks.
So, yep. What roles does circuit-wide EI play? The first way that we looked at this is by looking at the effect of different stimuli, contralateral versus ipsilateral. And you can see that if you just plot the amplitudes of the different stimuli, well, they're clustered, but they're clustered to the right of the sort of y equals x line. So in other words, they're all providing a kind of a net preference for contralateral responses, but they're differences. So S1FL here is much more active in contralateral stimulation than ipsilateral. And it is, of course, a major excitatory response. I didn't show you the histology for that, but it's true. In contrast, ZI, which I highlighted as an inhibitory input, is actually less activated in the contralateral response. So there is this shift, even though all of the responses remain positive, with the possible exception of this one right here, there is this shift. This APT region is a little bit more ambiguous, so we don't quite yet know how it figures. We also looked at the effect of adaptation. So very quickly, imagine looking at the stimulus, the response to a stimulus train early versus late in the train. The responses adapt. The late response is lower. And in S1FL, this is a pretty sharp effect. But in the presence of Gnostic, these inhibitory regions here don't adapt nearly as much. If we look at control animals that don't have Gnostic expressed, we don't see that effect. So what that means is that this failure to adapt in the inhibitory projections is due to the Gnostic probe. And we could calculate a ratio between the early versus late responses, and we could see that for these two inhibitory regions here, there is much less adaptation of the inhibitory signal. And so what we believe that tells us is that there is a circuit-wide contribution to the process of adaptation and also to the process of tuning for a stimulus, and that's what the graph on the left shows. So let me skip my future direction slide because I am talking more slowly than I thought. And just summarize quickly, I hope I've showed you that these Gnostic probes can report information flow in the brain, that projections transmit signals distinct from source region activity, that EI balance can be probed, and that it contributes to tuning and adaptation. In addition, there are many other things we can look at with these probes, and of course, we're also working on bioengineering to make the probes better, to avoid having to do differential imaging and so on. And I hope you can see how these results have some relevance to human brains, but you'll have to tell me. I think we've been able to look at principles that are pretty generalizable, but again, you tell me. And let me thank the people who did the work there. The people whose names are highlighted are the ones who contributed most. So thanks very much, and sorry to be slow.
Thanks a lot. It was great. It's super interesting. I think we will have maybe sometimes at the very end for some questions, and maybe if you agree, people could write some questions in the chat and you could already respond in the chat as we continue with the next talk, if it's all right.
Sure thing.
Okay, perfect. That's super interesting.
Yeah. I'm sorry, I can't see the time. I mean, that's one of the problems with the video way is that we can't see everything. Yeah.
All right. So I let Daniel introduce the next talk. If he wants, otherwise. Okay, well, okay, it's there. Well,
sorry, got a clinical call on the other line. Please go ahead.
All right. Well, I can do it if you want, Chris, or yeah.
Yeah, that sounds good.
All right. So as Chris said, Andrea could not be here, so we have her postdoc, right, Daniel, who will tell us everything about multiverse analysis. So please.
Yep.
Thanks a lot.
Yep. So I hope you can see my screen now. Yep. Okay. So yeah, thank you for having me here. So my name is Daniel Kristanto. I'm a postdoc in the University of Oldenburg with Andrea Hildebrandt. Yeah, unfortunately, she's not able to come or to have a talk today. So I will try to present her slide here. Okay, so this talk is about the multiverse analysis in graph-based fMRI research. So I will start with the problem statement. So we know that in fMRI research, the goal is actually to use the fMRI data to understand the individual differences in cognitive or in behavior. And one of the approach is to calculate the graph measures. So using a graph theory approach used in fMRI data or in a board signal. And to do this, we start with fMRI raw data, and then we do some kind of preprocessing, structural or functional preprocessing, we do some cleaning, and then we come into the functional connectivity, which is telling us the correlation between the time series of different brain areas. And once we get the functional connectivity, we can then treat this functional connectivity or treat the brain as a network or as a graph, where the nodes are the brain areas and the connections between them or the edges are actually the functional connectivity or the connections. So it means like if two brain areas they have stronger edges, so it means their functional connectivity are stronger. And once we have this kind of brain network, and then we can calculate some different features. For example, the global efficiency that tells us how information flow from one area to another area, or we can also calculate like a small wordness, local efficiency, and so on. And then once we have these kind of features, then we can correlate this feature or associate the features with the behavioral measures to understand how these brain features, the graph measures, explain the individual differences. And as I mentioned before, to come from the raw images of fMRI data to these graph measures or the brain network features, there are a lot of steps that is going on there. And there is a study published in 2020. So this study actually showed that different preprocessing steps taken in fMRI data may lead to different results. So this study here, they sent exactly the same fMRI data to 70 different teams and asked them to use the data to answer the same set of hypotheses. And these teams, they use different preprocessing steps, and then some of them come up with different research findings about the hypothesis. So it further emphasized that, yeah, there are a lot of things going on between raw data and then functional connectivity and also the graph measures, and different analytical decisions between them may lead to different results. And it led to an effort to do some kind of a robustness analysis check. So robustness basically to test whether the findings hold with different analytical decisions. However, for an exhaustive robustness analysis, knowledge of the full space of analytical options is essential. So this is where the multiverse analysis is coming. So it's kind of more systematic of robustness check. So for example, here, the traditional analysis, it starts from the data and then it comes from different steps. For example, the first step is data exclusion there, and there are actually different options there to exclude different data. But in traditional analysis, only one option in this step is taken. And then also similarly to the other steps like filtering or epoching, for example, for EEG. And in multiverse analysis, we consider the possible options for each step. So we started from the data there, and it comes to different options there. And then in the second step also comes to different options and also the other different steps. And then it will give us different results from different pipelines, or we can call it also different universe. So universe one, it means that we are taking the option one from step one, and then option one from step two, and option one from step two, and so on. And universe two, universe three, and so on. So this is the example of a multiverse analysis. However, in fMRI, there are a lot of things to consider to pre-process data, because we need to pre-process in the structural images. We also need to pre-process the functional images. We need to do some cleaning, and we need also to define the atlas or the brain parcellation, and we need also to define how we want to calculate the functional connectivity, what kind of graph measures we want to use, and so on. And what we need, if we want to do the multiverse analysis, that we have to have the knowledge space, or we have to have the list of the possible steps and the possible options that we want to take. So that's where our study come from. So we did this systematic literature review from 252 fMRI studies that perform graph analysis to associate graph measures to behaviors or to different cognitive scores. And from these 252 papers, we extracted how they pre-process their fMRI data, what kind of steps they are doing, and then what kind of option in different steps they took, and so on. And it came up, we found that actually from these 252 studies, we found that there are 61 different steps of reprocessing fMRI data from raw images to come into the graph measures from the functional connectivity. And these steps are visualized as a network here. So where the nodes there, where the cycles are actually different steps. And they are grouped into different groups, right? For example, the software here, and we have also the steps for structural preprocessing. We have also steps for functional preprocessing. We have also steps for noise removal, functional connectivity definition, and also for the graph analysis. So basically, these are the steps. And the bigger the steps, it means that these steps are used by more studies. And then the ages between those steps here, or the links between steps, it tells us that the study take this step and then other steps. So it's like the connection between one step and another step. And the thicker the ages means that more studies are taken this connection of steps. And actually, if we put some threshold on these networks. So here is example of we only visualize the steps or the network with the ages used at least by 33 articles. So starting from quite abstract and quite complicated network here, but if we put a threshold here, we can see actually some kind of commonality that across these 252 studies, there are some common things, some common pipelines to preprocess fMRI data. And from this kind of findings about the steps, we also provide some kind of analysis about the degree of each step. So the degree is basically telling us about the variability of other steps connected to specific step. So for example, if one step has a higher degree, so it means across these 252 studies, these steps are connected to different other steps. In the other way, if a step has a lowest degree or has a lower degree, so it means this step is connected a uniform other steps. So it's not connected to many different steps, but it's quite consistent across studies or across literature. We also perform some different analysis about the time, also how the changes of the preprocessing pipeline, fMRI preprocessing pipeline from two different times here. And what I would like to show here is we aside from the static figures that we have in our paper, we also provide some kind of interactive visualization of this finding that I would like to show here. So this kind of a app that you can access through the link that I showed you before on the PowerPoint presentation. And there are different nodes or different features on this visualization here. So the first one is actually the database. So if you click on the database here, it will give you some kind of the prisma diagram about the list about how we conducted the systematic literature review on what kind of studies that we included or what we excluded and so on. And there are also the included articles there, and also we have also the list of the steps. So there are 61 steps that we identified from the studies, and you can see the list here. And we can also see the list of parameters. The parameters here, it means the options for the steps. So from the 61 steps, we identified 17 steps that have variable options that are reported by the study. So for example, software, we have here different kind of software, and also we have the step of, for example, the temporal filtering. We have also different option of temporal filtering, or the steps of brain parcellation. We have also the option of different parcellation there. So we have the list of steps and also the list of parameters or the list of the options there. And another feature here that we can see is the steps there. So this is basically the graph representation that I showed you before, but this is more dynamic because here you can explore, like for example, you can click for each of the node, which is each of the step there, and then it will tell you what is this step and how many studies use this step and so on. And there are also other features related to steps. For example, here it will tell you like how many studies use different steps there. So you can specify the step that you are interested in, for example, alignment, and then it will tell you which studies actually taking these steps from our database or literature review. And there are also combinations. So combination is basically telling you how many studies use the combination of steps. So you can specify the steps, and then you can see the other steps and how different studies, how many number of studies use these two steps in combination. And similarly, you can also visualize the order of different steps. And the next features that this visualization has is in the parameters level or in the options level. So for example, you can define the steps here that has option or that has parameters. For example, here I would say spatial smoothing. So here you can see that this is the distribution of the options of the spatial smoothing. So for example, here 17 studies use 4 millimeter as a kernel, as the size of the kernel for spatial smoothing. And then you can specify the number of kernel here, like 4 millimeter, and then it will tell you which study is actually using this option for these steps. And you can also explore each of the individual articles. So if you are interested in one of the article, then you can just explore what kind of steps they use and also what kind of the options that they use in this of individual article. And the last features of this visualization is your own pipeline. So I found this one is, I think, the most interesting one in this visualization, because here you can specify your own pipeline. So for example, I would like specify this way. So just to make it fast, I just specify my pipeline has 2 steps here, and this is the steps. The first, I initiate the software, and then after I initiate the software, I would like to cut some number of initial volume, and this is the parameters that I have in my preprocessing pipeline. And when you click count here, then this will tell you how many number of studies use exactly the same pipelines as your pipeline. So here it tells me that there are 43 studies or 43 articles that use pipeline exactly as mine. And this also gives you the list of the studies that using the same pipeline as what you inputted there. And I will go back to my presentation. Yep. Okay, so that's our first work. So our first work is basically having this knowledge space about different pre-processing step in fMRI, especially doing the graph analysis. And the next one is that how to analyze the multiverse, because we know before that actually there are steps that you need to consider, and also there are a lot of options. And when you want to do the multiverse, it may be very computationally expensive, because then these different steps, also different options, also different ways to use them in different order or different combinations can lead to large number of pipelines. So exploring this pipeline, doing multiverse analysis on this large number of pipelines is really challenging. So there is an approach published in 2022 by Taflon. So the goal of this approach is to explore multiple analysis pipelines in multiverse analysis setting without exhaustive sampling. So the idea is that you can get the result of all the pipelines, but only doing the analysis on the subset of the pipelines. So the steps, there are two steps actually in this one. Well, I will just give you the general idea, but you can go to the paper if you want to get the technical details. So the step one is that you have to create a lower dimensional space to illustrate similarity between pipelines. So let's say, for example, you want to find the association between global efficiency of brain network and age, for example. But actually, to calculate the global efficiency, you can use different steps because you start from the raw fMRI data, you have different ways to preprocess your data, you have different ways to define your graph or your brain network. And then it comes, for example, you have 500 pipelines to calculate global efficiency. And then the idea of the similarity metric is that you get this kind of a 2D matrix where each dot is actually pipeline. And the closer dot, when the dots are closer to each other, it means that these pipelines are more similar to each other. So that's the idea of having this lower dimensional space to measure the similarity between pipelines and then to see how they are in a 2D space. The closer they are, the more similar they are. And once you have this 2D space, then you can apply the active learning to approximate the prediction profile of all pipelines by only applying to a subset of the pipelines. So from this 2D space, you have 500 dots there. The idea is that you only visit 50 of the dots there, and then you calculate the prediction performance of these 50 pipelines, for example, to predict the age from global efficiency. And then by only visiting these 50 pipelines, then you can get the idea or you can get the information of what is the performance of other pipelines without doing the prediction using the other pipelines. So that's the idea of this active learning here, using only specific number or using only the subset of the pipelines in the multiverse analysis, but then you can get result of the whole pipelines that you have in your analysis. And in our next work, we extend this active learning approach to also include latent variable as outcomes. So not only predicting observed variable like age or other observed variables, but we also make the active learning approach to be also connected or to enable it to predict also the latent variables. And we also implement the similarity for both brain-wide and also region-specific graph measures. So not only using the global efficiency as a global measure or a brain-wide measures, but also you can use then node-specific measures, like for example, the local clustering coefficient or the degrees and so on. And this is also another work that use also the knowledge space that we have, the graph of the preprocessing steps that we have. So the idea of this is that when you are doing the active learning, as I mentioned before, the first step is actually you need to calculate the similarity matrix. And to calculate this similarity matrix, you still need to do the different preprocessing pipelines that may also take a lot of times. So the idea of this work is actually to use the network of the pipeline, the network of the preprocessing steps that we have from literature review to mimic this similarity between pipelines. So instead, you run the pipelines, you get the output, and then you can calculate the similarity between pipelines. We just need the information of what steps you take for your pipelines, and then from that information only, we can say whether your pipelines are similar to each other or not without running them. So just by looking at what steps they are taking. Well, the result is not yet very, very good, but I think this is a promising direction because then we can calculate the similarity between pipelines by just looking at the steps what the pipelines are taking. And for the outlook, so funding has been secured to ensure the sustainability of the knowledge space. So basically, the literature review that I showed you before, it was humanly coded. So all the information from different papers we extracted by human. So it was it was quite challenging because for one study it took at least 1 hour, I think, to extract the pre-processing steps. So the next step would be something to use and fine-tune open source large language model. So for example, ChatGPT or BrainGPT here to optimize the extraction and the synthesis of data analytic decision. So whenever there is a paper, then we just use the LLM to extract the pre-processing steps, and then the human work would be only to review whether the extracted pipelines or the extracted steps by the LLMs correct or not. So it will definitely help to save time and also save resources there. And also, we also want to create and implement a template for reporting analytical decision in fMRI research. So basically, this also based on our findings when we are extracting different preprocessing pipelines. We saw that actually how they are reporting their steps of reprocessing fMRI data are quite variable across each other. And also, there is also, because we already started our second project on using LLMs, and we see also that LLMs, it has different performance to extract different steps. So it is also related to how papers use a different standard, or they report using different ways of how they reprocess the fMRI data. So this also our outlook to create such templates. So we know that this is also a community effort, and we aim to apply for an international network funding. So if you are interested to participate and to contribute, please send an email to Andrea Hildebrandt here. And yeah, I think that's all. And this is just another work also that we currently doing in our project. Also, we also develop a Python toolbox for doing multiverse analysis, especially in dynamic functional connectivity. So thank you very much.
Thanks a lot. It was really a great project. I can just imagine the amount of work behind it. So in the interest of time, I would suggest to keep the questions at the end, also for this talk, if it's okay. All right. Yeah. And if you can write in the chat the link to the... Perfect. Thanks a lot. And I'll let Daniel introduce the next speaker. Thanks a lot.
Well, thank you very much, Daniel. We've had two great talks now on the subjects of biological and statistical validation. And now it's my pleasure to introduce Zhen-Qi Liu, who will be speaking within these realms of biological and statistical validation, discussing PET and complementary and differing modalities. Thank you, Zhen-Qi, and I'll give you the floor.
Thank you. So I'll share my screen. Okay, so can you see my screen now?
Yes.
Yeah.
We see it.
Yeah. So hi everyone, I'm Zhen-Qi from Network Neuroscience Lab at the Montreal Neurological Institute. And today I'm going to share with you some of our recent progress on understanding functional connectivity. So this is probably just one of the analytical steps in the multiverse of the previous talk. And I'm going to show you that this is really, it is already overwhelmingly complex, and indeed, like, more attention should be paid into like understanding the analytical variability. So the contents are based on the this paper, and you can find the relevant code and data on GitHub. I'll start by asking, what is functional connectivity? So most people from my generation probably first learned functional connectivity as the brain networks, a kind of brain network. The brain is somehow acting as a network, and there is the structural and functional networks. However, the first impression might also lead to biases. So unlike structural connectivity, which follows actual physical tracks or pathways, there is no simple ground truth for functional connectivity. And we construct the functional networks entirely from statistical estimation. And the most popular way to do this is probably Pearson's correlation, but it does not have to be that. You're probably more familiar with some spectral measures if you are doing EEG or MEG. And people have been using and inventing many measures. And this seminal paper by Stephen Smith is just one of the many attempts to study the FC measures people were using at the time. And as a result, the functional networks can be very different. It also poses a challenge that which criteria should we use to evaluate them? these are the questions we're trying to answer in this project. So estimating functional connectivity boils down to measuring the relationship between two variables. The idea is actually more broadly embedded in many other scientific disciplines as well as the pairwise interaction. So we use this PySpy toolbox developed by Dr. Ben Fulcher and his group. It allows us to calculate hundreds of different pairwise interaction statistics, including many common use measures such as correlation, distance, mutual information, as well as the more obscure measures from information theory, from causalities, and so on. And to give you an idea of what measures we use to calculate functional connectivity, I'm showing all the pairwise interaction measures we benchmarked in this paper. And the numbers after the measures are the number of variations belonging to that measure. So in total, for these 49 measures, we have 239 statistics. And in the main results, we calculated all of them for 326 unrelated subjects from HCP Jasanoff dataset. And each subject has 4 resting state rounds. We're using the Schaefer 100-node parcellation for this. So with the 239 FC matrices for each subject, we first ask how similar or different are they? And we quantify this by showing the edgewise similarities between them. What I'm showing here is a similarity matrix with both rows and columns being the FC measures. You can see that they're highly organized and form clusters that follow their categories. As an example, the conventional Pearson's correlation is in the covariance family, which is in the top left dark red corner here. And if we follow it down column-wise, it has a positive correlation with the covariance family itself, negative correlation with precision, distance, and strong positive and negative correlations with some of the information theory measures, and not a lot of correlation with most of the spectral measures. And here I'm showing some of the group average FC matrices. The intuition is just to show that they're quite different just by looking at their appearance. Then naturally, we would imagine that they would have very different topological and geometric features that we commonly measure. So we start by showing just the distribution of the edge weights. The 239 measures are side by side on the x-axis. The darker line suggests there is a greater number of edges for that FC measure. So you can see some densities are highly skewed, while others are somehow more evenly distributed, suggesting differences in their topological organization. For example, this might lead to network hubs. And here I'm showing the weighted degree or the commonly measured hubness of the different FC measures. While some of the measures have the typical pattern with hubs in dorsal attention, ventral attention, or the unimodal visual and supermodal networks, there is also considerable variability across the measures, with some of them showing more of a spatially distributed hubs, emphasizing, in contrast, the transmodal regions. And next, we check to what extent each of the pairwise statistics capture these two very well-studied features of functional networks. The first is the negative correlation with inter-regional Euclidean distance. And overall, we can see most measures are either positively or negatively moderately correlated with distance. There are several measures, however, they have a pretty weak relationship. This also corresponds to the other feature we are going to study, which is structure-function correspondence. We would expect a relatively strong positive correlation with structural connectivity. Here it's estimated from diffusion MRI. And for most of them, this is true, but still there are some of them having pretty low correspondence. This suggests that even for these very well-known properties, the results strongly depend on the choice of the FC measures. And after this more of a topological features, we next consider how they would more directly reflect the underlying biology. And here we quantify how different types of FC correspond to their other biologically meaningful interregional networks. And in specific, we have these correlating expressions from the allen-human brain atlas. We have Larmian similarity from big brain cell staining data. We have neurotransmitter receptor similarity from multiple PET tracers. We have electrophysiological connectivity from the resting state MEG data, and finally metabolic connectivity from dynamic FDG PET. And we're asking how well each FC matrix aligns with the networks estimated as all those different spatial and temporal scales. And in general, we observe the strongest correspondence with neurotransmitter receptor similarity and electrophysiological connectivity. Perhaps counterintuitively, the correlation with metabolic connectivity is not that strong, suggesting there is a different amount of shared variances. We also find that across the FC measures, covariance or correlation and Pearson and precision measures are pretty good in terms of correspondence, but things are quite different for some of the information theory and frequency measures. So after studying this more of a group level results, we next turn to some of the individual level features. And we want to first test how good they can be used to tell apart individuals for these different FC measures. This is a probability analysis, also known as fingerprinting. And we calculated the identifiability index here and found that so precision, which partial correlation belongs to, really stands out in this one. This is a bit surprising, but also is consistent with many previous reports of how this partial correlation might be better at prediction and telling between the individuals. Meanwhile, Pearson's correlation or covariance in general is pretty good in this case. Next, another test in quantifying individual differences is we consider how different FC measures can be used to carry out a generic prediction of individual differences in cognition behavior. And for simplicity, we just followed the approaches from Ken and colleagues and used IC-derived these 5 components as a target. We use ridge regression to predict the component scores for each individual from their individual FC measures. And across the cognitive behavioral terms, we see the overall magnitude is pretty consistent with the previous reports with some noticeable outliers. And it seems the pairwise statistics that perform well for individual fingerprinting are also kind of performing well for the cognition and behavior prediction. And this says that it's probably important to select the appropriate FC measure for a specific task, such as brain behavior prediction here. So now we've seen how FC measures relate to other types of interregional matrices and these external phenotypes. We turn to FC themselves and try to ask if we could provide some mechanistic explanation. And back to where we wanted to quantify the relationship between the 2 time series, we can think of them as 2 blobs holding the information they contain. And now to study these 2 blobs of information, one way is to check their overlap and getting the common unique parts or shared variances. It's related to the intuition behind the mutual information, but the framework we're using is much more general than this. So with this partial information decomposition framework, we can decompose the information between these 2 variables into 4 atoms. And I'm showing here in the 4 colors in the Venn diagram. And a more intuitive demonstration is on the right, and the 4 atoms are unique 1, unique 2, synergy, and redundancy. The unique ones are pretty easy to understand, and what's more interesting are the other 2. So synergy means when 2 sources of information are considered together, they provide something new that they cannot be retrieved from either source individually. And I'm using the yin-yang pattern here to illustrate this. And in contrast, redundancy means each source provides the exact same copy of information as the other. So when they are combined, they will be repetitive. And now we are actually going one step beyond this because we are more interested in the information flow between the 2 variables. So back to the Venn diagram, we can replace the labels with the 4 information atoms. And the simplest way to think about information flow is probably we just give them a past and a future state. So now information transfer happens between past and future. We have this in total 16 possible transition patterns. And this approach is known as integrated information decomposition, or FID. The 16 information flow patterns may be intuitively understood as information storage, information migration, information decryption, encryption, and others. And this can be mapped by this pairwise correspondence matrix shown here. And the theory guaranteeing this to work is actually quite complex, but there have been many interesting results and insights deriving from this. And Dr. Andrea Hildebrandt has some excellent papers if you want to learn more about this framework. You can also refer to these two original papers about the formal series, information series behind FID and PID. So here we have the 16 atoms as the matrices. Actually, you may notice that for the first one, which is the redundancy to redundancy flow, this is quite similar to what we would expect from a common Pearson's correlation calculated functional connectivity, and that is indeed the case. And by the way, here is an open source toolbox you can use to calculate these measures. So to see how our FC measures are represented by these information flow patterns, we constructed some simple linear models and estimated the contribution of each of the 16 patterns to each of the FC matrices. So the rows are the 16 patterns, and within each column, the darker or lighter shows their relative contribution. We find that for the classical statistics such as covariance, precision, and mutual information, they most reflect the pattern where redundant information stays as redundant, and this is both reassuring and kind of boring in a way. But some of the spectrum statistics predominantly reflecting unique to unique the information flows. This is a pattern where information that is exclusively stored by one region stays as exclusive to that region. In both cases, they will belong to the pattern of information storage, where information is kept the same over time. But there is this much more diverse information flow patterns than this. For example, some of the information theory and causality measures, maybe they are specifically designed to capture the different information flow patterns. These show the transfer of synergistic information belonging to encryption and decryption, or more formally known, they are also known as downward and upward causation. And there is a lot hidden in here, and actually we did not try to interpret each one of them. We're trying to, in general, convey the idea that there exists a full spectrum of information flow patterns that are where yet to be explored. And this is not possible with the commonly used Pearson's correlation, and we might want to specifically targeting the capturing of such information. And finally, I want to summarize this paper by providing some conclusions and recommendations. And in this paper, we see that different statistics give very different FC matrices, and there is substantial variability even for these very well-known properties. And this means FC measure is quite depending on the context. This also alerts us that there is not necessarily a single best measure, but actually different measures targeting different mechanisms. And with no ground truth can be very tricky and also in the meantime important to pick an FC measure, and it depends strongly on the research question you're asking. So we have some general recommendations here. So what should we do? Well, first, you should always match the measure to the experimental question. In our findings, we do find covariance, distance, or Pearson's correlation. They are in general pretty robust. So there's a reason they're being widely used so commonly in the research community. We also find precision or partial correlation has some desirable features in some of the scenarios, such as individual differences and predicting brain behavior relationships. And finally, maybe it's possible to combine or engineer specific FC measures to get some specific sensitivity or precision purposes, such as capturing distinct information flows. And as for future directions, I think first, as you can see, there exists a lot of biological complexity in measuring the FC, and different modalities, FC methods, they may capture different aspects of it. And there's a lot more to explore here. I really want to recommend this review paper about advancing FC from association to causation. The fact that we're seeing partial correlation standing out in some of the cases we had is a great example also highlighted in this paper. Okay, that's all for my presentation. I'd like to thank my lab mates and collaborators. And you can read the paper here. And here's also how to get in touch with me or the lab if you have any questions. And thank you all for listening.
Thank you. Very good talk. I appreciate it. That now leads us, I think, to our fourth talk. We've heard a lot of very great things concerning biological, statistical validation, and now it's my pleasure to introduce our colleague, Matej Perovnik, who will be discussing in more detail clinical validation, and I'm very happy to cede the floor to him.
Thank you, Daniel, for the introduction. Hope you can see the slides. Okay, so after a bit more broader talks, I'll focus on the clinical sides, and I'll focus just on one methodology, but focus many pathologies. So to start it, I mean, we listened a lot about biomarker validation. I'd just like to share this figure from a paper which I like. So conceptually, when a biomarker is further described, we go through a phase of development. The biomarker is then described in one sample. It's cross-validated. Then we go into the second step of prospective validation, generalization, and lastly, if everything goes according to plan, but as you can see from these hypothetical scenarios, many biomarkers fail along the way. We go to population level and maybe achieve research or clinical utility. The methodology I'll present is called scaled subprofile model, which is based on principal component analysis. The majority of work using this methodology was done using FDG-PET, but as I'll show later, it was also extended to resting state fMRI. In the first step, we obtain our data, in this case, FDG-PET scans from diseased and healthy individuals. The scans are spatially normalized and smoothed. Then, after obtaining control and diseased individuals, in this case, showing individuals with Parkinson's disease, the data is transformed to subject by voxel data matrix. We extract the gray matter by thresholding or masking. Next step is logarithmic transformation and double centering by subtracting the row and column means. And then a principal component analysis is applied to obtain so-called subject score vectors and associated eigenvalues and principal components. Among those principal components, one or combination of several principal components can be chosen as disease-related pattern. And this is done so that the top PC or several principal components are retained. And they represent the network that best separates the two groups. So in this case, patients with Parkinson's disease and control individuals. The methodology can then be used on a single patient level. So if we use the pattern and a single subject scan, transforming this into row vectors by multiplying one with another, we obtain a scalar or subject score, which can be then compared with a reference population. And in clinical practice or in research setting, we can see if this topography or disease-related network is present or expressed in a certain individual. Now I'll go a bit back in history to show you how this was all done, and lastly I'll present some of our current work. So going first showing Parkinson's disease individuals, because this was the first pattern that was described. as said in the introduction, we begin with one sample, healthy individuals, individuals with Parkinson's disease, identifying the pattern. Yes, Shin? Okay. Identifying the pattern, so this was our candidate for a biomarker. Then, in the next step, we want to see if this biomarker is also present in new populations, so not the ones included that were used to derive this pattern. So the next study that I'm showing here from 1999 included 4 sites, more healthy individuals and patients with Parkinson's disease. And we can see that the expression of this pattern was replicated in different sites. So simply put, patients with Parkinson's disease had higher expression of this pattern. In clinical utility, we are not only interested if one biomarker can separate disease from healthy individuals, because that's relatively, or can be thought a relatively easy task for a clinician, but we want to know if a certain disease is characterized by this biomarker and not other similar diseases. I'm showing here, or showcasing one such paper in which the authors studied the expression of this pattern, now called Parkinson's disease-related pattern, or abbreviated PDRP, so disease-related pattern, in individuals with different types of dystonia. Shown in the graph here, we can see that patients with Parkinson's disease express the pattern, but not individuals with dystonia. Another important step, now we are in the second box of validation of biomarker, is a so-called test-retest. And here, several studies tried to replicate this pattern topography by newly identifying the pattern and trying to correlate the topography with the existing one. Then we can also ask ourselves if this topography is changing, if the patient is scanned on one day or after a day or after a month. I'm showcasing here a study in which it was shown that Parkinson's disease-related pattern, because it's important that a biomarker is stable and can be replicated in various sessions, so it's not specific on the time of scanning, and it does not changes from day to day. And it can be seen here that within session, using radioactive water PET, the topography is highly stable, but also between session. So patients scanned one day after a day or after 2 months. And here it's important that the time between scanning is chosen so that the disease pathology, and this is different for different diseases, does not meaningfully affect the brains in the meantime. And we can correlate the topography of the patterns, or we can correlate, so on one hand topography, or on the other hand we can correlate the subject scores. Showcasing here that the subject scores obtained with radioactive water PET or FDG PET are highly correlated on a single subject level. What about replication across labs? This was also done in the case of PDRP. This is one of the study because this pattern has been replicated in several labs. This is one of the study from our center in Ljubljana, Slovenia, where we recruited patients with Parkinson's disease. We identified the pattern, so this is the first step. And then in the prospective validation step, we have shown that the pattern expression scores are high in patients with Parkinson's disease, but not in patients with atypical Parkinsonian syndrome. So these are the diseases that can clinically resemble Parkinson's disease, but have different underlying pathology and, more important, different prognosis. Comparing patterns that were identified across different labs. So in previous slide I've shown it's important that patterns can be replicated across sessions. Here we can see that pattern identified in our cohort highly correlates both on the topographical voxel level in patient to the pattern correlated identified in the US cohort and also on the single subject score level. And my colleague also showed that because moving biomarker, in this case PDRP, using different scanners, you can either try to make every setting as close as possible across sites, or try to see if the biomarker is robust enough. So the different, for example, in this case, reconstruction algorithms, it was shown that they have minimal effect on the pattern expression, which is a good characteristic for a biomarker that could be used across different clinical sites. So lastly, in regard to the PDRP, shown here is a slide that, because I have highlighted several of the studies showing how these steps were undertaken for the PDRP pattern, summarizing here in this paper, we can see that this pattern has been identified not just in one, but several cohorts across worldwide, rendering that this is a good biomarker for the Parkinson's disease. Furthermore, more validation in a clinical setting. It is important that on one hand, a biomarker is stable so that it doesn't change from day to day or from month to month. But we want to have a biomarker that could track disease progression and could correlate with clinical scores, showcasing this study in which we can see that the expression of the pattern increases linearly in years. And as mentioned before, more recently, the SSM-PCA model was modified a bit to try to extract or to try to see if a similar pattern topography can be also extracted from resting state fMRI. And showcasing here, without going into all the modification of the method, we can see here that the pattern that was described as a functional PDRP, highlighted by regions in cerebellum, primary motor cortex, prefrontal regions, basal ganglia, can also be used to discriminate patients with Parkinson's disease from healthy controls. And looking at the graph on the upper right part, we can see that the two patterns are highly comparable. So either using crusty state or FDG PET in differentiating Parkinson's disease patient with from healthy controls. So how about clinical utility? One common question in clinical routine, as mentioned before, is differential diagnosis. And showcasing here, because these patterns can be used on a single subject basis, we can obtain FDG PET scans from a single individual. And I didn't show all the patterns, but this steps of biomarker identification, validation, replication across labs can be also undertaken for other, in this case, Parkinsonian syndromes. And using the methodology, we can calculate not just one, but the expression of several different disease-related patterns. And in this case, we can calculate the expression of Parkinson's disease-related pattern, which I've talked a lot about in previous slides. Expression of and then in the upper two rows, we have topography of patterns of two atypical Parkinsonian syndromes, which clinically resembles Parkinson's disease. So this is a real life example in which one patient was scanned. All the patterns the pattern expression scores were calculated alongside with a pattern that correlates with cognitive decline in Parkinson's disease. And then we obtain in clinical practice subject scores, which are z-scaled according to the normal reference population. And we can use a simple logistic regression algorithm to try to calculate the probability that the patient belongs to one of the three classes pre-specified in the algorithm. So either the patient has Parkinson's disease or atypical Parkinsonian syndrome. This was a patient that during life was difficult to diagnose, but the algorithm made a very strongly supported diagnostic decision. And the patient was then follow up until his death, and the suspicion of Parkinson's disease, as indicated also by the algorithm, was confirmed in the pathology. And here I'm showcasing one patient, but this has been replicated on a larger cohort of clinically difficult to diagnose patients with Parkinsonian syndromes, and it had been shown that the algorithm, which is based on the expression of these patterns, can outperform early clinical judgment in difficult cases of Parkinsonian syndromes, in which the clinical features can be in the beginning mild and difficult to differentiate between different diseases. And as mentioned, other neurodegenerative disorders, not just Parkinson's disease and atypical Parkinsonian syndromes, but also neurodegenerative dementias. These steps have been undertaken in the years of research to show that this is something that can be used and can be replicated in clinical setting. Not just for differential diagnosis, but also, for example, for tracking disease progression, shown in this study, in which we included individuals with mild cognitive impairment and healthy controls. And we have shown that those patients that converted, so the CMCI group over here, that converted from mild cognitive impairment, which is a prodromal state of dementia due to Alzheimer's disease, have an increased expression of a pattern that is characteristic for Alzheimer's disease. And this can already be seen in cognitively normal individuals, which later develop cognitive decline. Similar, in Parkinson's disease, we have prodromal clinical stages, and it has been shown that Parkinson's disease scores can predict time to conversion in patients with so-called REM sleep behavior disorder. This is a disorder in which patients only have sleeping problems, and it's not known how fast or in which of several diseases they will convert. And a study from colleagues in Freiburg showed that higher expressions of a certain score, here they used the cognitive pattern in Parkinson's disease, predict shorter survival in patients with dementia due to Parkinson's disease. moving to the second part of my talk. I'll introduce dementia with Lewy bodies now. This is dementia, which is the second most common neurodegenerative dementia after Alzheimer's disease. And it's characterized by memory or by cognitive problems on one hand and by Parkinsonian symptoms on the other hand. And also for dementia with Lewy bodies, we have in the first step tried to define the pattern, identify the pattern. So this is graph over here. This is the pattern topography. The regions shown in blue are hypometabolic, and they are correlated or connected with regions highlighted in red which represent hyper metabolic regions. So this was the pattern identification group and internal validation. So patients from one cohort from one scanner. The pattern was identified also by other groups in Japan, Korea, and the Netherlands. And we can see here just eyeballing, but we can also correlate the patterns topography that this topography can be replicated in various clinical groups. Let's try to validate the pattern, so to see if the changes that we observed in our small cohort can also be replicated across a larger cohort of a multicenter sample. In the study that has been just recently accepted, we included almost 400 patients with dementia with Lewy bodies, and extending the cohort with a small subset of patients with a prodromal stage, so patients with very mild symptoms that may develop dementia with Lewy bodies in the following years. In addition to compare, so I'm highlighting once again the need for differential diagnosis, not just diagnosis between disease and healthy individuals. We obtain data from patients with dementia due to Alzheimer's disease, and so the MCI cognitive impaired patients, and this group was then subset into two small groups based on the presence of alpha-synuclein co-pathology. The alpha-synuclein is a main driver in dementia with Lewy bodies, but it can also be present in certain individuals with Alzheimer's disease. So the study was set to have a derivation and internal validation set. So this was already shown in the previous slide. Then we had an external single testing data set and an external multicenter testing data set, in which we included a multicenter sample of patients with dementia with Lewy bodies. So this is what was already shown in the before, and then we calculated the expression of this pattern in a prospective settings in patients with dementia with Lewy bodies and healthy controls, and we can see that across the internal validation, but also in external single testings and external multicenter testing groups, the expression of this pattern is highly significantly elevated compared to healthy controls. And as mentioned before, it's also important to see if this expression means anything in relation to clinical scores, and we can see weak to modest correlation with UPDRS score, which is a measurement of motor impairment, and MMSE scores, which is a measurement of cognitive impairment.
A couple minutes more left.
Okay. Lastly, extending this to the MCI cohorts, we can see that the pattern is already expressed in patients with MCI due to Lewy bodies as compared to healthy controls. And I'll skip this for the discussion. And we can see, as shown before for Parkinson's disease, that the expression of a DLB pattern is increased linearly across years. So these were patients that were scanned at multiple time points. And it's already seen in the prodromal stage. So this is 0, and this is the reference point. So that the pattern is also already expressed in the prodromal disease stages, as shown also in the graph on the upper right. So to summarize, I hope I have showed enough evidence from more than 30 years of research that position SSM-PCA as a well-established method for identification of these disease-related patterns or networks, and that now we have a lot of evidence that disease-related patterns can be used for diagnosis, differential diagnosis, tracking disease progression, following treatment response in those diseases in which treatment is available, and to provide prognostic value. And this method has been recently also introduced by the official guidelines, clinical guidelines in that specify how to read FDG-PET images as one of the semi-quantitative analytical methods that can be used in clinical practice. And PDRP is listed one of the PD biomarkers in the novel biological classification scheme. I'd like to acknowledge all the people that contributed to my research I presented in the last part of the talk, and everybody that contributed to years of research in the last 30 years. And thank you for your attention.
Well, thank you, Matej. That was terrific. We've now had all the talks spanning our interests from statistical, biological, and clinical validation of molecular connectivity at this point. And what we'd like to do is open up the floor for sort of a panel, and anyone that has particular questions they might want to raise, please just raise your hand, and we can begin a discussion.
Okay,
Chris.
I just had one really concrete question for Matej. The FDRP, so the functional MRI related Parkinson's disease related pattern, did that use variability data, or what's called amplitude of low frequency fluctuations, or was it done in a different way using the time series and connectivity?
So the first step was done using ELF.
Okay.
May I?
Yeah, just go ahead. Nobody else is there then.
All right, so I have a few questions for Alan. So, if I understand correctly, one of the messages is that when we see, our very simple positive correlation between two regions, what could be behind it could be basically anything. Is it?
Yeah, maybe not anything, but yes, some complexity. Yeah.
So, and in particular, you mentioned the excitation to inhibition ratio as a possible source of, thing that might vary across different projections, different regions. So my question is, I don't know if you gave it some talk, like how can we understand in humans what is at the basis of the connections that we observe? Because I know that, for example, with EEG, there are some ways to compute an E to I ratio based on the slope, I think, of the power spectrum. But I don't know, did you see any way to translate these to humans, essentially?
It's kind of funny because I asked the same. I think it's a super hard question and maybe unfair question. I asked the same in the chat already internally.
Oh, sorry.
Yeah, but no, but I think it's super interesting. Yeah,
I mean,
that's very
I'm
nice.
super interested in it too, but it's a nut that, I can't say we've cracked. I mean, in part because we're still using these tools for the first time, or the second time, as the case may be, in animals. I think there are two ways that one could think about it. So the first is look at the way that I think we're thinking about it currently, which is where we're interested in circuit level phenomena in animals. How does the brain respond to a sensory stimulus? How does it adapt? How does it develop selectivity and so on? And then to look in humans for signatures that correspond to what we see in animals. So that's one way. But I think the way that you're probably thinking of, if I may, is that you see phenomena in humans and you want to understand how they, what they reflect in animals, and that is what I am not as strong at because I don't work in humans. But this is where I think having a meeting like this and maybe an offline talk might be pretty useful because it's simply not a direction that I've yet thought that much about. That said, I mean, I think there's some problems that that we can already look at. As I say, I feel like for a while, whenever I told people about the idea that we would be mapping information flow from one region to another, they said, they would say, well, we can just look at the connectivity from DTI and know what the source activity is, and just that's where that's going to tell us enough. And I think that's something that we actually refute. So it is relevant, but maybe not in a constructive way yet.
Yeah. Yeah, we
Yeah,
have
and
to
also,
talk. Yeah, sorry.
sorry, I don't want. Sorry, you go ahead.
No, just after the talk of Alan, I'm surprised that Matej and anybody else would find a biomarker that is stable across different conditions and diseases. So I mean, there is hope, even if we don't understand maybe what we see means, but we can still do something with it.
Yeah, I was going to say I don't want to editorialize too much, but these things that you did very nicely, I thought the laterality as a factor, as well as the frequency, different tags, all these dependencies. I mean, some of them could at least be empirically mapped out in human functional connectivity studies. Maybe they have. It just seems people are very comfortable using a signal that's just a convolution of a gazillion things, and then just go crazy and do liberal arts, almost analytic techniques, And I think maybe some of your manipulations, some of them could be tried with humans, right? Obviously not the sort of invasive ones, but stimulation parameters that are mapped and other things, in tasks.
May I ask another question if nobody else? Yeah, okay. Because I have a question both for Daniel and Zhen-Qi, or kind of it's complementary questions. Because to my understanding, what you did is that you look at all the different ways one can compute some graph theory metrics, but were they based all on Pearson correlation or on anything? And the same question for Zhen-Qi. So you look at very different measures to quantify connectivity, but did you do any attempts to do some multiverse analysis or try the effect of different pipelines? Because, based on what Daniel showed, every measure we use can be computed in many different ways. So it's complexity is expanding a lot.
Yep, that's true. So what we did is we map the options, but we did not try them empirically. So that's correct that actually there are different ways, for example, to calculate connectivity, but actually beyond that also, even before calculating connectivity, we have also different ways to preprocess the fMRI data structurally or functionally, and also to clean noise and so on. But I think that's also what we are going to do next. So we want to empirically test this kind of mapping, how well the answer that we want to get is not to get the best practice, but the answer is more toward which decision matter the most. So I think that's one of the important points that we would like to answer also next.
Yeah, that was a great question, and I think for us, we did not explicitly testing the multiverse of processing choices. We did indeed, in the supplementary, we tested for a few different data sets. They have different pre-processing choices. So we did find like the similarities between the different measures. They are generally pretty similar in terms of their relationship between the pairwise interaction. But the actual FC matrix, I'm sure they're kind of different. And also the similarity is high, but like there's space for the variability that is probably accounted for by the quality, the acquisition, and the pre-processing choices of the different data sets as well. Yeah.
So we need some multiverse analysis from now on. All right. Any last comment, questions, considerations? All right, should we wrap it up, Chris?
Yeah, I think we can wrap it up. Yeah. Thank you to all the speakers. I thought this was super interesting, and I really have to, my brain was stretched, particularly embedding of multiverse choices to really think about this. So I thought it was great. Thank you, everybody, all the speakers and all the people who attended. And I for the people who are not in our molecular connectivity working group, I think you're invited to attend these seminars. I think Xin showed a schedule. I think our website is. I think we can maybe, if you could just put it in the chat briefly, somebody. Hang on, let me find it. For people who are interested in what we're doing, who our members are, please visit molecular connectivity. Yeah, exactly, molecularconnectivity.com. Okay, so that's all from me. You have anything to say, Arianna and Daniel? And I think we'll let people go. I think, today we had a choice in enforcing strict discipline between having talks, limited to 20 minutes, but I thought it was good maybe to let people sort of speak and not interrupt, because it was so interesting.
They were too good. Sorry for being late, but
yeah. Okay, so I hope everybody has a great weekend, and thank you for showing up.
Thank you, everyone, for making it.
Thank you very much.
thanks a lot.
Thank you.
Bye.
Bye.